Yes, sir.
Yeah, we can proceed now, ma'am.
Okay, thank you. Welcome, ma'am.
Yes, thank you so much. Good afternoon to everyone.
Okay, so respected Deshmukh sir, President MSI India, Dr. Shilpa Jani ma'am, that is President MSI Gujarat and Goa State, Dr. Vijay Kumar, Regional Coordinator, Saurashtra and Kutch region, dear participants, academicians, researchers and students from all over the country, a very good afternoon to all of you in this fourth session of expert lecture series. Also, on behalf of one and all present here in the virtual meet, I would like to take privilege to welcome our eminent speaker, Dr. PRITHA Mukherjee. She is Assistant Professor in Medical Lab Technology, Dinabandhu Andrew Institute of Technology and Management from Calcutta. She has completed her PhD from University of Calcutta under Department of Biotechnology. She has expertise in various skills like cell culture techniques, tumor sample handling, animal handling, various molecular biology techniques, biochemical assays, enzymes, and metabolic assays, DNA isolation, transformation of cells, cloning, to list a few of them. Her area of research is in the molecular mechanism underlying cancer and exploring disease biology. She has published research and review articles in various reputed journals, and her research publication in recent is Scientific Reports in Nature Publication House. Other journals include Oncology Reports, Artificial Cell, Nanomedicines and Biotechnology, Journal of Biological Chemistry, Molecular Cancer Therapies, and a lot of them more. So now I request ma'am to deliver her expert lecture. Please, ma'am.
Thank you so much for the kind introduction. It was really a lot of things told about me. And first of all, I would like to express my sincere gratitude to my respected seniors and all the expert members who are present here. One of them is obviously the president of MSI, Professor Deshmukh. Sir has introduced me to the MSI platform, and every day I'm learning something new, interacting with people, because I'm quite young in this field. So I have a lot of endeavor to go ahead, to do forward. And avenues like this is not only going to help us interact with the young generation, with my colleagues, with my contemporary people, but it is also going to help us inculcate ideas so that we all can work on it and try to implement it in future. It is really a great honor for me to be present here in this expert series. I have heard the one or two lectures which were there before my lecture in the previous weeks, and I'm sure my sincere gratitude to RK University, to all the eminent members of the university present here, to Dr. Swati ma'am, with whom I've been interacting in the last few days, and she has been coordinating the entire thing. So RK University and the respected members who have been called for the series, it is a very nice launch, I must say. And I have also promoted it to students from my place. I have also told them that please go through this series, because you all are bringing up topics which are really very versatile and very varied. So now today is a Sunday afternoon. I would not do much ado. I know that people have just finished their lunch and they are here to sit and listen to something which is crisp, short, but interesting. So just before I begin my talk, I would like to share my screen. Swati ma'am, is that okay?
Yes, sir. Yes, ma'am.
So I actually come from a background of microbiology. I have done microbiology and biotechnology in my graduation and post-graduation. So there is a lot of attachment to this subject. I have
Ma'am,
tried because there is a lot of, yeah.
excuse me, ma'am. Sorry to disturb you, ma'am. Please go for the full screen.
Okay, I think there is some problem. I was trying to do this full screen thing, but this is not working. I don't know
Okay,
why.
okay, no problem, no problem. It is visible.
Okay, ma'am. Then okay.
You can proceed.
Visible.
If you
Yes,
cannot.
I was trying, sir, but I think something is wrong with the keyboard, but it is not happening.
Okay, okay.
It's okay, ma'am. Please continue.
If it is not visible, just tell me, Swati ma'am, then I will increase the zoom ratio a little bit more if it's needed.
No, no, it is visible. It is visible.
Okay, okay, sir. Okay, thank you. So actually, I come from a background where I have been working on microbiology and biotechnology were the subjects which I studied, and I do have a lot of interest for them. But in my PhD, I started to work with cancer biology as the main focus of my work, and it was related to the fact that we all know that there is a problem of cancer relapse in today's world. That people who are being treated with different chemotherapies or different radiotherapies, whatever it is, they are not getting absolutely cured. It is not a part of the clinicians, it is not a part of the researchers. It is still a mystery that what is happening inside the body and why are the cells being not absolutely targeted. So that was what I worked with, and there I found that cancer, though we call it, some people still debate that yes, it's a genetic disease, but of course there are many different factors which are responsible for this grave disease. Now at this current time, we are all worried about the pandemic. We are sitting back home, we are all locked up, and we are all very conversant with the coronavirus, the COVID-19, and it is of course it is again a microbe. It is again a microbe. It is a virus which is actually not letting us go in the proper mode of survival. Similarly, there are other diseases also which are not very insignificant. Like before this COVID-19 came into it struck the entire world, there were diseases which were more grave, which were more severe, and which were more pertinent. Now, cancer is one of them. So if we leave the genetic factor about cancer, there are still other factors which are influencing it. And it is not a surprise to us today. We all know the great discovery of Helicobacter pylori and how it is associated with gastric cancer and so on and so forth. So starting from there, I have taken today's topic like microbiota and humans. It is actually a mystery of love and hatred that continues. We know that nothing in this world is going to be absolutely the way we want, and nothing in this world is going to be absolutely different from the way we want. So it is actually in between, a line in between where we are standing, and we are not alone in this race. Why are we not alone? If we ask about the fact that why are we not alone, then just a glance into the different, there are several different magazines and several different scientific journals that bring up the fact that human body is not a single body as a whole, but it is a combination. It is like an ecosystem. And this ecosystem is made up of human genes as well as microbial genes. So the microbes reside in our human body, and we are all aware of the fact what we call the commensals, because people here are all from microbiology and biotechnology background. So I don't have to go into much detail about how the microbes are residing in our body, but just to draw the fact that this research is so interesting and so prominent that there are journal pages from the Economist, from the Scientific Americans, from Nature itself, who talk about the human microbiome. So it is not only the virus or the bacteria or the other microbes which are there in the environment, but it is also the part of the microbiota that is within us. And this microbiota is, of course, a more focus of the research in the current world, because this is the twig which you can change, or which we can look into, or this is the tweak which actually promotes the disease condition in the body. So if we have to understand several diseases which are caused by microbiota, we have to understand how they are residing in the system and what are the problems they can cause, and not only problems, they are beneficial too. So we have seen many cartoons where there is a microbe and there is a dialogue box which says we are not harmful. Yes, of course, they are not harmful. Some common people who are not aware of microbiology and biotechnology, who are not so privileged to get the advantage of, like us, like we studied, like all of you studied, maybe there are people who are not that privileged, they can call the microbes quote unquote germs. So when we go out in the road, in the streets, we talk with people and people will be like, there are germs. Yes, they are germs. They are germs because they are in the environment and they can affect us. But when we talk about the human system, then it is the human microbiota supraorganism. And why it is a supraorganism? Because we, the humans, are composed of, say, 23 to 25,000 genes are discovered. There are many more yet to be understood and to be discovered. But inside this, the microorganisms contribute to at least 3 million genes. So we can understand that there is a good correlation between the microbes and the humans, and together a human body is not a single human body alone, but it is a harbor of microorganism. So we call it a supraorganism, and when the environment affects the human body, it is not only the human body that is responding, it is also the hub of microbes that is responding to the environment. And there can be several interactions with respect to toxins, beneficial metabolites. Microbes can secrete some metabolites, can secrete some toxins, which can affect the human body. Simultaneously, the human body can give a resistance, say, because we have immunity, so it provides a resistance. Also, it can be give them a harbor to proliferate, to grow, depending on what kind of nutrition we take, what kind of environment we are facing. So this health status, the health and disease condition, mostly we remain in the transition between health and disease, and any kind of tweak in this microbial genes or the human genes can lead us to the disease condition if and when the condition permits us to do so. So when we talk about the microbes which are there in the human body, we all are very aware not only of the human genome. We all know that human genome has been widely studied and it is already known what are the genes and how they are placed. Of course, the functioning of some genes are being worked upon. There are people from genetics who are really doing hard work in discovering the interaction and the signaling and the work. But Human Microbiome Project was a big endeavor in this aspect. Like when Scientific American and Nature talk about human microbiome, they actually begin from the Microbiome Project, where it was not only told that our body can have different predominant microbial types, but this microbial types can vary depending on the location of the body. And we know about the natural microbiota, the commensals which are there, but it can differ depending on the different locations of the body. It can also differ based on the health and disease condition. And some microbes do have genetic similarity, some have genetic dissimilarity. Not only that, the most unique point of the microbiome project, this is the heat map, which shows the entire microbiome that is available. But it was also found that there can be loss and gain of functions at the individual depending on the strains which are inculcated. This is very important for researchers or for people who have interest in microbiology like us to notice that depending on the different environment we go, we can harbor or we can inculcate different strains. And those strains can actually help us or actually determine the loss and gain of functions that will be taking place in the entire microbiota that resides inside us. So when we talk about the microbiome and whatever we have seen till now, it is very clear that it is actually an intercommunication between the host and the microbiome. It is not only residing, it is not only quietly and silently sitting in our body, but it is always and continuously intercommunicating with us, that is the host. And this kind of intercommunication can be noted in several different organs, but a very important organ which there is a lot of work going on in this particular aspect is the gut microbiome. Particularly in the gut microbiota, if we see this, there are different microbes like the lactobacillus. In the duodenum, we have the streptococcus. In the distal ileum part, there is mainly a predominance of the Clostridium species. In the proximal lumen, again, there is lactobacillus. Then there is jejunum and colon. Colon has a predominance of bacteroids. So this is what is actually the general microbiota of the colon. But apart from that, this dominant microbial species not only reside over there, but they go and interact with the cells of the lumen. And if we are taking, suppose, for example, if they are in the lungs, or if they are in the gut, or if they are, say, in the other parts of the body, everywhere, there is an interaction. And that interaction actually not only brings the epithelial cells into connection, but also the immune cells into connection. So now a very interesting topic that is being worked upon is how immunotherapy can be modulated by using the bacterial toxins per se, or the bacterial metabolites, or the products which are released from the bacteria. So we can understand that this interaction is actually enforcing or enhancing our innate immunity. When it is enhancing our innate immunity, it is playing a role in the immunological domain. But apart from that, it is also carrying out several signaling procedures which are important for maintenance of the cells, for growth, for proliferation, for cell division, etc. So constantly there is a competition for the allocation of space of the microbes, and there is colonization. And the amount of colonization again can influence how it is interacting with the host. So now the question is, we understood, okay, the microbiome is there. We have a second genome. All of us have a second genome, and the second genome is the microbiome. So we have a second genome. The microbiota is residing inside us. It is definitely not sitting quietly, interacting at each and every moment. But what are the different methods that can be used? Yes, of course, there has been a revolution in the development of microbiome and the different methods for study. A very important domain is the study of the DNA, because even if we are not being able to identify the microbe which is there, if we can look into the DNA, then we can identify into which species or into which category you can segregate it into. So nucleic acid research is a very important part of understanding the microbial community. Apart from that, we also have work that is going on under the RNA, protein, and the metabolite domain. I just have a slide later on where we'll see that the microbiome is now interacting with the metabolome. So any part of the body is not left alone. Even with the metabolome, till today it was as Deshmukh sir was also discussing, like the metagenomics platform is very important. Swati ma'am will be knowing much better than me in this regard, since she's working on that. So DNA, metagenomics, and the transcriptomics and proteomics part is definitely very important for identifying a community. But again, now it is also not only restricted to the DNA, RNA, that is not only to the central dogma, but also to the metabolomics. And we know that mitochondria itself is an organ which has evolved, which has co-evolved. So human was never independent. Human is never independent still. Always there was a concept of co-evolution. Say you started from the mitochondria, And today we see that, of course, we are not independent because we are associated with a second genome. Now, this mitochondria itself is the hub of the not only of ATP production, but of the different metabolic sectors that are going on inside our body, like the different carbohydrate metabolism, say per se, the lipid metabolisms, even the transporters. We know some of us must be knowing about the different transporters, like what we eat. Suppose we are taking up glucose. Glucose is being channelized in the body because of the GLUT transporters. Now, these GLUT transporters or glucose transporters are a part of our metabolism. But these glucose transporters are very well linked to the cancer relapse also. Just, I would like to mention one thing over here, because since I have seen that particular thing in my PhD life, what I have seen is people who are treated with chemotherapy, they have very high expression of the drug efflux pumps. Now we know drug efflux pumps, just for the basic, drug efflux pumps are kind of pumps which are on the surface of the cells, on the cell membrane. And their work is not only to take up, they can take up drugs, they can also efflux out drugs. Now, when the cancer tumors are being treated with chemotherapy, the level of the drug efflux pumps are increasing rapidly. And these drug efflux pumps, so you can understand, they will be effluxing out the drugs from the tumor cells or from the cancer cells. And therefore, some population of the cancer cells will be there who will never be affected with the drugs. And that population particularly resides within the tumor. And suppose you withdraw the chemotherapy from the patient after, say, 5 or 6 months, and let the patient to lead a normal life for, say, again, another 6 months, you will see after 1 year, the patient comes back reporting a case of relapse. So why is this relapse happening? A major factor is those cancer cells, particularly we call it the stem cells now. Some of you must be very aware of how the stem cells are affecting the different treatments. So if we leave about stem cells, just consider it as a population of cancer cell X, which is having very high drug efflux pumps, letting out the drugs and not allowing chemotherapy or radiotherapy to be fruitful or to be active. So similarly, these pumps, like the ABCG2 ATPase pumps or the B-type ATPase, these pumps are again very well correlated with the metabolomic pumps, that is the GLUT transporters. So nothing in our system, you can leave it isolated. Everything, the main thing which I want to impart from today's talk is that it is not a one single signaling done system or one signal organ done system. It is actually a system where the different components are interacting with each other, and any fault or any defect in one can simultaneously go on and so forth affect the other normal functioning of our body. So apart from that, in order to study the microbiome, there are also several techniques like we see in the right hand side of the box. There are techniques like different clinical trials. Different clinical trials are being carried out to study if the different microbiome replacement therapy is working or not. Apart from that, there are germ-free animals that are being taken up in the transplantation studies in lab. Also, there are several intervention studies with the fecal microbiota transplant therapy, which is FMT. I will come into that slide in the next one. Yes. So microbiota sample analysis and therapy designing. Here, what we are seeing is like from a stool sample. This is like you can go from the stool to the statistics. This is very interesting. So you start from a fecal sample of a particular person, and then from there you can do the genome extraction and bacterial genome sequencing and follow it by different bioinformatic analysis with the different softwares where you can identify the species, analyze the diversity, how different it is from the original report of the receptor patient, also analyze the abundance and function of the species that you are getting. And from here, we can isolate the particular fecal microbiota, We can wash the rest of the particles with antibiotic if it is not necessary or if it is hampering the system, and take the fecal microbiota and restore it into the particular healthy, either in the healthy donor or in the healthy recipient. So that what happens, this microbiota can itself go and reform the microbiome in the system. If there is certain problem in the microbiome, or if the microbiome is not similar to a healthy patient, then we can do fecal microbiota transplantation therapy. This is being used nowadays. Some part of it is already in the clinical trial, but a lot of studies are going on in order to do this identification part, like how can we categorize and how can we group when we take it from a particular sample. So this microbiome in our body is being influenced by several factors. Our day-to-day factors, as we saw in the initial slides, every factor, say the environment or the amount of stress or the medication that we are taking, and of course our genetic composition is going to have an influence on the microbiome. Not only that, the diet that we are eating, what kind of diet we follow in our system, like we know Western diet and Indian diet will be different. Not only that, if a person is taking, say, plant-based diet, then it is seen that there is more of bacteroides microbiota in their gut. Compared to a patient who is compared to any person who is taking, like, say, more of oil or more of animal protein, that person will be having more of firmicutes like species in their gut. So depending on how we are living our life, how we are interacting, what kind of medications we are taking, our gut microbiota will be affected. And this gut microbiota, now we can say, okay, so what happened? It is only the gut microbiota that is being affected. The other part is normal. But no, now there are very interesting studies which are talking about the gut-brain axis. We all know about it. We have heard that gut-brain axis, there are studies who are telling that it is actually the microbiota of the gut which is influencing our brain activity. Brain activity in the sense, if we just pinpoint it to be more specific, it is mainly the neurotransmitter level. So there are studies which have shown that particularly in our area itself, like in West Bengal, I was associated with one study where we were trying to find out that people, that the problem of arsenic in this Delta Basin is very high. In West Bengal, Bangladesh, this part has a very high deposit of arsenic. Now, there were reports in the government sectors which told that this arsenic affected basin in this part, you have an increased rate of suicidal tendencies. Now, why is that? It is absolutely no way related. Suicidal tendency is a psychiatric disorder, and arsenic is something related to water, which is going into our system. How is this related? Very interestingly, the study is not yet published, so I would not go into the detail, but very interestingly, some of the initial studies that we were doing, we found that the level of neurotransmitters like the serotonin, the dopamine, they are very different in the people who are having a high ingestion of arsenic. We also depicted it in the mice model, and we are trying to establish some mice models for this study. Hopefully it will be done, I'm not sure, but it is difficult to analyze this kind of models, but the problem or the discovery of this gut-brain axis is now very strong, and whatever is there in the gut, that is influencing our brain. So what kind of environment we are in, what kind of stress we take, what are the medications, like there are some, if a person is on alzolam, if a person is on some kind of sedation due to some neurological disorders, definitely that is going to have a different impact on the gut microbiota, on the commensals, and obviously that is going to indicate the brain to function in a different way. So these are different areas where we can see how they are influencing our microbiome. Now, this microbiome is however, it is important to a health condition, but definitely this microbiome can also implicate some kind of disorders. Disorders in the sense, Particularly in this picture, I have noted a few disorders like the neurodegenerative, the psychiatric, of course, the problem of cancer, which we will talk about in a very brief in the next few slides. And there are cases of inflammatory bowel disorders, which comes from the problem of the gut microbiota, the metabolic disorders, which I was talking about, the like obesity, like the different problem of all the different problem of the enzymes and the hormones that we have. And also, of course, infection cannot be left out, because infection is the most common phenomenon which we consider in case of disorders by microbiota or the microbiome. So particularly, just a little look into or zoom into the hallmarks of cancer and how the microbial derived signals can modulate this cancer through the different mechanisms. A very excellent paper by Ellman in PLOS in 2017. It's a very recent work, and if you go into the paper, you will see that very well they describe the fact about, we know about the hallmarks of cancer by Weinberg. So that is already known. But how these hallmarks can be twisted or these hallmarks can be targeted by the different microbiota, particularly if we talk about the intestinal microbiota, then it is going to help in resisting the cell death. It is going to suppress, it is going to obliterate the growth suppressors. Growth suppressors, as we know, P53, PI3 kinase, these are the main genes that we look into. Then enabling the replicative mortality also, they will increase the mortality, they will cause genome instability and mutation. They can promote some kind of inflammation in the microenvironment, and they will, of course, create a immune surveillance. When they create an immune surveillance, the immune destruction, the immune cells will not be able to identify the cancer cells, and they will not undergo natural destruction. So that is what we mean as the immune destruction, avoiding immune destruction or creating a surveillance mechanism. There are different researches which have been done in the current years, and they all talk about the different intestinal bacteria, like the fusobacterium, like the enterococcus, we know the bifidobacterium. These are some common names which we all are used to, which we all know. But these bacterial intestinal bacteria can actually target some specific hallmarks of cancer, and this has been established by our different PDX, by our different xenograft models or mouse models as described in this table. Apart from that, now it will be a question that whether it is only the gut that is being affected, and what about the other parts? No. There are definitely the extra gut organs, not only the gut microbiota, but of course the liver, the brain, and the lungs. These also are hub of different microbiome, and this microbiome or microbiota that is residing in these organs again are part of very specific immunological signaling. That is, as I mentioned, there can be different metabolites which are secreted or excreted from this microbial organisms which are residing in these particular organs, and they can help in homing in different immunological parameters. Like we know about the TLR signaling, example of Kupffer cells which are there in liver. Kupffer cells can be a target of this deviation from the normal. And apart from that, the NK cells, the Th17 regulatory cytokines, and also if we come into the brain, then it is mainly the microglial homeostasis that it is being focused into. If there is a deviation, the homeostasis is lost. And we've already talked about the gut-brain axis, which is very important. In the lungs too, we'll see that lungs definitely with aging, again, this is another important parameter. With aging, there is again a change in the microbiota. Now, this change in the microbiota with aging is responsible for either increasing or decreasing the immunity as per in what condition that person is residing. That can be particularly by, say, the interferon signaling, or by the Th17 cytokines, and also by the presence of, sometimes by the presence of the myeloid cells. So as I was telling, the lung microbiome can induce carcinogenesis via causing DNA damage. So what are the different probable causes? One of them is, of course, aging. But apart from that, it can integrate the DNA damage into the host. It can cause inflammatory response alterations. There can be some chromosome instability, abnormal signaling pathways, which can be triggered by either defect in the immune system or due to some toxins that are being released. And definitely that is going to affect the homeostasis of the system. So from a healthy lung to a lung cancer situation, the microbiome can play an important role in carrying out these particular deviations, which are actually bifurcations of the one and same problem. And in this figure also we see how the lung microbiome can, okay, I see a red strike over here. I think that was due to somebody had mistakenly shared. So can I
Hi.
delete?
Yes, sir, it is. Yes, ma'am, it is here. Maybe Shahid, one of the participants, he must not be knowing the thing, and by mistake, it is like here written.
Yes. So okay, so I think I've been able to clear it out.
Yeah.
Yes, ma'am. Yes, ma'am. Yes.
Yes.
Thank you, ma'am.
Yeah, thank you. So again, the same thing is being described here, like carcinogenesis and homeostasis. These are the opposite sides of balance, you can say. Either you have homeostasis condition, or if it is disturbed, then it can lead to carcinogenesis. Of course, there are other conditions, there are COPD, etc. Definitely, it's not possible to club everything into one talk. So just let's focus on carcinogenesis and genomic instability. So in between that, what is there lying in between these two is the lung microbiome. So lung microbiome is exactly in between these two phases. And how the microbiome is influencing the condition is actually determining whether it is a lung homeostasis or there is a probable way of leading it to carcinogenesis. And my work was particularly on breast cancer. So I have taken samples from breast cancer patients during their MRM surgeries. And I looked into the different signaling mechanisms which are imparting chemoresistance. But I have a specific interest of knowing and also working later on with eminent scientists or whoever is interested in this microbiome. I definitely, because during my research, I found that yes, breast cancer, the microbiota of the breast is in normal and tumor condition, the microbiota can change. Like if we have if we say have a tumor condition, there can be relative abundance of some different kind of bacteria in the system compared to the ones if there is normal. And all these kind of bacterial organisms which are residing in this particular, in the mammary fat pad or in the arterioles and ductales of the mammary gland, they can again be identified by the 16S rRNA sequencing. Uniquely, they can be identified, they can be grouped into several clusters. And also, there are studies which are talking about which particular microbe is implementing a particular metabolite or say a particular toxin, which is leading to this problem of disturbance in the microbiome population. And that is why the patients who are having breast cancer, they are showing a very unique and different microbiome compared to the one which is normal. There is a definite alteration in this particular site of tumor. Also, another important, like a cancer which is again very much into notice nowadays, and there is a predominance in case of women, particularly not only breast cancer, but this cervical cancer is also in a very predominant condition. And in case of cervical cancer, it has been seen that from normal to viral infection, like we know HPV is a major cause of the cervical cancer, and there are people who have isolated the virus from the specimens, from the cancer samples, and they have studied into whether they are mutated or not. If mutated, then what kind of mutation is there in the viral sequence, and how is it actually hampering the normal healthy tissues of the system? So during this study, important parameter which was found is that in the course of the infection, or in the course from dysplasia to invasive cancer, from dysplasia to metastasis, there is a change in the composition of the microbiota. So we can understand that the microbiome which is there in our body, it is being affected by the environment. And when it is being affected by the environment, it is undergoing a change. And that change is actually implicating a prominent mark in our host system. Like for cervical cancer, it was mainly the increase of microbes, a decrease in the dominance of the lactobacillus, increase in the level of anaerobes particularly, and of course some mutational characteristic changes in the HPV. So this interaction is not a single interaction. Definitely there are loops, and one interaction is influencing the other, because maybe it is beginning in the gut, but the metabolites are being released in the blood, and from the blood it is going to the different harbors. Say again, the skin has got a very huge hub of microbiota. So the skin is also influencing this metabolite secretion, and then we can have a direct effect of the formation of tumor from the skin microbiota or from the metabolites, or it can be coming from a different angle through the metabolites in the blood, which can also be influenced by diet, and also that can trigger the immune cells. So together it is a big loop where this entire microbiome and the relation of cancer, whether our body is loving the microbiome. If the body is loving the microbiome, then it is going to keep it and it is going to be beneficial. And if the body starts to that same microbiome, which actually the body was in love with once upon a time, it can start to hate the same microbiome when that goes out of context, and that starts to secrete metabolite, and then again dysfunctions the immune system of our body. And as told that in order to evaluate the loops, we can look into different factors. One of the factor, as we have discussed, metastasis. Another factor is inflammation. Inflammation, because inflammation is very well connected to the release of the different cytokines, the TLRs and the NF-kappa beta, and also reactive oxygen species. Definitely this ROS is actually in the center of, say, any kind of hypoplasia or any kind of proliferative or inflammatory diseases, because in cancer there are still a lot of work going on, yet the balance of this ROS, what is the level of ROS and how are the antioxidants in our body trying to balance it? This is still a puzzle and a mystery. And the more number of research works you go through, you will see that there are different schools which talk about this in different way. Because ROS is no doubt a very, very important causal route for different inflammatory diseases, be it the inflammatory bowel syndrome, be it the lung inflammation, be it the gut inflammation, or be it some kind of inflammatory release in the blood. Whatever it is, ROS is a in the center of the entire thing, you can say. And this ROS is affected by different Colibacter, different E. coli species, and this dysbiosis, the dysbiosis that it can cause in the gut, that can ultimately lead to different diseases. One of them is definitely, again, cancer. And so the microbiome is affecting the cancer. How is it affecting? As in not only inflammation, but definitely there are cases of primary to distal tumor metastasis. Like suppose the tumor was in the gut, but later on when the patient comes back and reports cases of relapse, you can see that there are traces found in the lung, traces found in the liver, traces found in the bone, or when it spreads to the bone marrow, then it is absolutely dangerous. So this kind of metastasis to the important organs of the body, say it liver metastasis or say lung metastasis, this is again, microbes are like a channel for that metastasis probably. Like the fusobacterium, it can travel from the primary tumor to the distant site. And the Fusobacterium species in this particular study, I've taken from a paper which where you see this insects are actually the Fusobacterium histology part and histology of the tissue. And it was seen that the same Fusobacterium, which was there in the primary site in a patient, that patient comes back with cases of relapse, you will find the occurrence of the similar Fusobacterium and similar metabolites in the distal metastatic site, say it's the liver or the lungs. So you can confirm that yes, there the origin is the microbiota or the microbial organism, which is transferring from the primary to the distal site. Again, a very interesting study showed that when we migrate to US, when a person migrates from the US to the US in India, in these Asian countries, here we have a high microbial diversity. So there we have an enriched metabolic function, and this is actually the useful part that we are talking about. So this microbiota is helping in the metabolic functions of our body. But again, if we migrate to the US, suddenly if there is a change in the place where we are residing, then it can influence the gut microbiota. The gut microbiota, the diversity can be decreased per se, and there could be some depletion in the metabolic activity, in the metabolic function. And that now we don't know. Maybe that depletion can again lead to some kind of a difference in the intestinal microbiome, and maybe that is why the incidence of cancer, again, if we just correlate, incidence of cancer in the West is much higher than what we have in the Asian countries. So as I was saying, this is another parameter, the microbiome and the metabolome can be connected. Particularly, we see in the level of the metabolic receptors or the aryl hydrocarbon receptors. These aryl hydrocarbon receptors, they promote the Treg cells. And now we know that when we all know about the problem of the HIV. HIV is again a disease which is caused by the, again, influence of the microorganism. But that particular disease is due to the defect in the immune checkpoints and also the T cells, to be more precise. So these T cells, the lymphoid T cells and the intestinal T cells, the Tregs, the Tregs are, apart from the B cell and T cell and the CD8 and CD4, the Tregs are another important group of T cells which are actually helpful in regulating the immunological background, say in the level of cytokines, and also in promoting immunological balance. Now these Tregs are the metabolic receptors, can be regulating these Tregs, and these metabolic receptors can have a direct effect on the anaerobic respiration, or just if we say it in the reverse way, the dysbiosis or anaerobic respiration caused by the microbes, which are like Clostridia and the bacteroids and the Enterobacteriaceae in the gut, in the intestinal mucosa, they can go and induce a different metabolome signaling in the metabolic receptors. And that can again lead to different production of hypochlorite, nitric oxide in the system. And that is again correlated with the INOS and the NOx genes. And there are studies where we know that INOS and NOx, they are important genes which are responsible for the inflammation, for uncontrolled proliferation, etc. So here we see that this interaction can take place in different levels. Not only it is not a single level interaction. It is in steps. It is in multiple steps. The microbiota can first interact with the mucosa, then it can go and invade the lumen, and then it can interact with the digesta part. And so and after the digesta part, it can go to the colon. From colon, again, there can be some secretions which can go and influence the immunity parameter. So it is not a single step reaction, but of course, there are multiple steps in which this microbiota is going to interact with the human body. And then in this course of interaction, it can cause dysbiosis or disbalance and give rise to neoplastic cells. In order to study, like, how will we study these different steps and how the steps are being channelized? So some kind of interaction studies can be found out by particularly by the sequencing platforms and also by PCR and analysis. But these are the software generated the plots which tell us in which particular chromosome you can find the effect of that of the change of the 16S rRNA. If the microbiota is changing, then in which particular human chromosome can it affect, or if so, then whether it is in the cervical or colorectal or bile duct, where the predominance, where you can find the more of the predominance of that particular microbe. And definitely, if our body is interacting with the microbe, the immune system cannot be left out from this, because it is actually the immune system which is trying to combat the microbe in the first place when it is going to tweak. Now, in order to combat the immune system, in order to combat the microbe, the immune system is, of course, whether it can get activated or it can be suppressed initially. Now, whether it is getting activated or whether it is being suppressed, this change is being generated by the microbial modulation again. So the microbiota in our body is actually affecting this huge crosstalk of immune system that is lying. And of course, if you can modulate the microbiota, then definitely you can alter the immune system, and that can lead to generation of a healthy condition in a human. Microbiome engineering has been done recently, and it has been shown that if the microbiota can be engineered, then it can reduce the pain reception strategy in the cancer patient. So because pain reception is again connected with the brain, gut-brain axis. So a different engineering, or if the microbiota, particularly if they have worked on the colonic microbiota. So colonic microbiota, if there is a modulation done in the microbiota, they have seen it in the xenograft models in mice, that the perception of pain can be reduced. And that is again a good part of the microbial engineering, or since we have the microbiota, we can do this engineering. So that is what is the advantageous part of how you can put the inherent microbiota to some use. Put some alteration therapy, we have to design some modulation therapy so that it can help in restoring the balance in our system. And definitely, the diverse microbiome has shown that many cancer therapies like the chemotherapy or the pelvic irradiation or radiotherapy per se can damage the healthy microbes. So if the engineering of the microbes can actually lead to some kind of balance restoration, definitely we must also look into the fact that how the therapies are going to affect the system. If the therapies are going, there are case reports which say that yes, the chemotherapy or PIDs or the radiations can definitely damage the healthy microbe. If so, we have to supplement these therapies with some kind of fecal microbiota transplant therapies or some kind of microbial modulation therapies, so that the healthy microbiome is restored and the subsequent damages do not take place. So definitely we know the name of probiotics, and since we are talking about therapy and how to restore and whether the cancer patients, they have to be treated with some kind of other therapies later on so that their microbiota suffices. Yes, apart from FMT or fecal microbial transplant, the prebiotics and diet, that is an important factor. Also the probiotics. Probiotics will be having a good implication in the anti-cancer therapy, no doubt, because the probiotics and the FMT together can help in restoring the dysbiosis condition. Like if there is an inflammation in the gut, and there if we see that tumors are being originated as a result of this microbiota and the mucus associated with it, which is going awry, we can definitely put into use some kind of probiotics and FMT, which can actually not only enhance the effect of the different treatments so that the treatments can be more targeted and more proper, but it can also reduce inflammation. It can reduce gut dysbiosis and secondary conditions like diarrhea, like vomiting, etc. So that if you can control the inflammatory parameter, then definitely again the problem of the excess progression of cells or the problem with the different immunological parameters or tumor mass development can be controlled. Similarly, I will not go into the detail of this slide again, this is the same image where they are showing that what are the different therapies which you can conjointly use. And these, all the therapies are nothing but based on the microbiota population itself. So we see that microbiota is influencing the disease condition. Again, microbiota is responsible for influencing the homeostatic condition. So if there is a disease condition imparted already, we have to check for what are the healthy metabolites that the microbiota can secrete, either supplement with those metabolites to mimic the condition that yes, the patient has the healthy microbiome, or you supplement it with a microbial therapy, modulation therapy itself, or some kind of probiotics and prebiotics or symbiotics, which can work in conjunction to reduce or enhance the levels as per requirement. Like if we see beta glucuronidase, these are important for the functioning of the lumen. So the anti-cancer drugs will have a side effect. They can reduce the level of these enzymes. So we can supplement it with these particular enzymes. We can also supplement it with different chemotherapeutic or different probiotic agents, so that the main concept is the diversity is not lost, but the target should be to restore the diversity. That is what is important. If we can restore the diversity, then the metabolome will be functioning, the glucose metabolism, the fatty acid metabolism, the immune functioning, the everything will be in proper check. Also, the endocrine functioning of certain organs is again correlated, like we were saying, the level of the hormones, that is also correlated with microbiome. So restore the diversity even if there is a loss due to the different treatments that are probably harsh on the body. And just to conclude, these are the last part of today's discussion. So we talked about bacterial microbiota in general, and what can microbiota secrete, what how they if it's inside the body, how they can act, whether they can modulate the immune system or the metabolome or the diversity or the cell cycle, cell division. But very interestingly, these is now put into these bacteria, they are put into a very different use now, like nanotechnology. There are we all know about how nanotechnology is advancing, and particularly in one of my work, I saw that in breast tumors in mice, when I developed mammary tumors in mice and treated them with nano formulation of different drugs, particularly say vedolactone. Vedolactone is a cumestin drug. It's a cumestin drug originally, but we made nanoparticles of that. Similarly, temozolomide, we all know it is a very important drug, which is for the functioning of the different glioblastoma, for restoring the function of the glioblastoma cases. So temozolomide nanoparticles, vedolactone nanoparticles. Also, we had designed some nanoparticles with proper, say, like modifications on the surface, so that they can go and target the breast cancer cells particularly. So these, I have seen that yes, nanoparticles do have a very targeted effect, and the advantage is that since they are in a very small amount and they are enclosed in a shell, they do not impart bifurcated toxicity. They do not go and affect the normal cells, and in a very particular environment, like if you can make it pH sensitive, then a particular pH itself, it will be released very slowly into the tumor, into the origin of tumor, or into the tumor site, so that the effect is long lasting. So these are advantages of nanoparticles. But now what is being done is there is a little bit more engineering that is working into the system. The bacterial effector nanoparticles are being designed. That is, in case of Yersinia pestis, this left-hand side, this image, this shows the Yersinia pestis. They have taken a particular metabolite from the Yersinia pestis and they have conjugated it in the nanoparticle. And they have seen that if you compare the no treatment versus the treatment case, the bacterial effector coated nanoparticle treatment, the number of dead cells is increasing. When the breast cancer cells are being targeted, the dead cells are increasing, and the survivability of the breast cancer cells is decreasing, which means it is a better prognosis. It can lead to a better prognosis in case of a tumor patient. This is another example of the same type of therapy where we say that it is bacteriotherapy being done. Very interestingly, bacteriotherapy is being combined with it is again an example of breast cancer therapy, where for breast cancer therapy, they combined bacterial therapy along with breast cancer therapy. And it was seen that definitely the tumor specific, there was a homing of the tumor specific E. coli cells into the particular site of tumor, and that could lead to degradation or say damage to the cancer cells. So this study was done in mice, and this is also it is published, but there are again many works which are going to endorse this study. There are many different avenues which are still to be left, but just wanted to give you an idea of how this bacterial study, how the microbiome can be studied, how the microbiome is useful, how it can lead to damage conditions. If damaged, then what are the different recent techniques that are upcoming, where students, if there are students in this platform, I know, so it is mainly an encouragement notice for the students. This is an avenue which is definitely very interesting, needs a lot of research and needs a lot of good and all fertile brains like you. And this area particularly of designing nanoparticle delivery of drugs into the system to cure disease, how you can use the different bacterial effectors to help particular, sufficient, more targeted and more foolproof delivery. So this is what it was all about today. Thank you so much.
Thank you, ma'am. Ma'am,